Hi everyone, today we're going to talk about human leukocyte antigens because I've got an issue and it's about tissues. If you look at the cell on the page here, you can see that there are antigens on the surface of it. Antigens being markers that help it distinguish if it's a friend or a foe, a self or a foreign cell. These antigens have a special name. These are human leukocyte antigens that are found on the surface of almost every cell in your body. These HLAs help the body recognize which cells in your body are yours and which ones are not. So if a cell has my HLA, even if it's from someone else, my body will think it's my cell. Conversely, if a cell has HLAs that my immune system has not recognized all this time, then my immune system will designate it as foreign and then attack it. So obviously HLAs are super important because they must match the donor's cells with the recipient's cells in their immune system. The donor cells that they have on the tissue that they are donating must be recognized as self cells even though they're coming from a different source. So what we need to do is we need to do a test called tissue typing or HLA typing. This test is going to determine how similar the antigens on my cells, the recipient cells, are compared to the donor cells. Now, the closer that these HLA antigens are on the transplanted organ matching with the recipient means that the more likely that recipient's body will not reject the transplant. Rejection meaning that the immune system attacks it. There is no perfect match. Even with all of these tissues matching up with their antigens, even if the immune system sees it as much self as possible, the patient will have to suppress their immune system. We are just trying to figure out how much to suppress that immune system with immunosuppressant drugs so that this person doesn't get sick from germs because we're suppressing their immune system too. So we want as many of these HLAs to match with the organ donor as the organ recipient. Now, the HLA's expression, the generation of these HLAs, both in the shape and the number of these HLAs that are expressed, is controlled by a set of genes located on chromosome 6. We call these set of genes the major histocompatibility complex. You get two versions of this MHC, the major histocompatibility complex, because you get two versions of chromosome 6, one from mom and one from dad. So that means you have two different alleles of the same set of genes that is going to generate these antigens on the surface of your cells. And these antigens are generated if they are active enough to be synthesized from the genes that tell them to do so. The crazy thing about this is that each version, each allele that you get has 6 different kinds of HLAs produced. So if you have 2 different alleles, one from mom, one from dad, you have the potential of 12 different antigens that are expressed in each and every one of your cells. Now, not every one of your cells is going to express every single one of these genes, but they have the potential. They're all there. You have tons of different antigens, and therefore, this can get kind of complicated. We'll focus mostly on 2 main classes of these HLA antigens. Every human is going to inherit one antigen from each patient, so you'll get one version of the HLA, one version of the HLB, one version of the CW, one version of HLA-DR, one version of the DQ, and one version of the DP, where my picture is in the way. That's DP right there. When performing this typing test for a kidney transplant in particular, like our patients in our example, we're going to look only at the HLA antigens of the HLA-A, HLA-B, and HLA-DR because those are the only 3 that are present on the surface of kidneys. The problem still is that these major histocompatibility complex genes are among the most polymorphic known to medicine. As in, when we think about genes that express different traits, think about your eye color, for example. We have people that have brown eyes, people that have green eyes, people that have blue eyes, people that have hazel eyes, so on and so forth. And that's not just you have it or you're not. That's, hey, I have one trait versus another versus another versus another. That's what we call polymorphic. These antigens that are being produced are also polymorphic. There are dozens and dozens of different kinds of these alleles that can be expressed for each of these sites. So each allele has to be identified by a number. And you can see here, HLA-A1 or HLA-2 are just two versions, two alleles that are possible at the HLA-A site. And you can see that there are dozens and dozens of those available, and they're very complicated in naming. The large number of these possible variations and combinations of these HLAs make it really hard to find just a match out of thin air. No one that is unrelated to you should have these same HLAs displayed. They shouldn't have the same genes, and therefore they shouldn't be expressed in the same way. But you do have people who are very closely related in genetics to you, your family, which makes your family a much more likely match than the general public, because you at least got some of your genes from your father and some of your genes from your mother, and so did all your siblings. Or some of these genes might be similar to your aunt's genes or uncle's genes, and you might be able to match with your cousins a little bit easier. By analyzing which 6 of these HLA antigens both the donor and the recipient have, we are able to determine the closeness of the tissue matching. What would be ideal is if all 6 of those different HLA antigens generated were a match between the donor and the recipient. That would be ideal. The likelihood of that happening is a 25% chance, because you might have one version. Let's say this is you right here. You might have this version from your mother and this version from your father, whereas your sibling could have entirely different version from your mother and the same version from your father, or another sibling might have the other version, etc., etc. This 25% chance is very, very, very small, but at least because it's a partial match with mother and father, because there's an opportunity for the mother to pass down the same chromosome to each child, at least there's a possibility that you have matching genetics with a donor and recipient, being between sister, brother, or likewise, or cousins even. Now, traditionally, HLA typing was done using serological techniques. They would pull the blood from the patient and mix it with a serum containing antibodies to determine which antigens were present. If they had a agglutination inside that sample, if they had the antibodies on top of the antigens, then that means that there was a positive test, and therefore there would be the ability for us to know exactly what the tissue typing is based on the interaction between the antibody and the antigen in lab. Now, HLA typing is done primarily using molecular techniques. We're talking about DNA sequencing. We'll isolate the DNA, we'll amplify it with PCR, duh. We do that with almost every genetic test we do. And if we sequence them to determine the particular allele present, then we can know who's a match just based on the sequence of DNA rather than the expression of the antigens and using antibodies in lab, which can be expensive once you get as many antibodies as there are for all those tissues. Plus, it's very time consuming. This is just generated by a computer. Once the donor and the recipient have been tested for compatibility, the next step is actually called an antibody screening, which we also call a PRA, or a panel reactive antibody. The reason we need to do one more test is that we need to figure out how many different HLA antibodies are actually expressed. You can express a ton of that same one or very little of the same one, meaning that if I have antigens all over my cells, my organs might not be very good to give to someone else because I would have my tissues targeted much easier with all those antigens on the outside by the recipient's antibodies. So what we're going to do is we're going to take a small amount of the organ recipient's serum, the stuff inside their blood, mixed with cells from about 60 different potential donors. And each of these tests is done separately, so it's going to be in a well, just like you did with the microarray. We'll see if those antibodies in your recipient's serum are going to target those cells in those 60 different people. And if they do, that means that they have a high potential to target any cells. So if a patient, say, has 30 of 60 reactions in these little wells, they have a 50% reactive antibody. Their antibodies are reacting to half of this huge population of people. So they have a 50% PRA. The lower the number, the better. The lower the number, the more likely they are to, or the less likely they are to reject a transplant, because that means their antibodies are less reactive to other tissues, even if they aren't a perfect match. After the tissue typing is done, there's going to be some screening to conduct called a cross-match test, meaning that we're going to figure out once and for all what will happen to the cells that we insert into the person's body based on the HLA antibodies that you generate. So a small amount of the potential donor's white blood cells, that's the person's leukocytes, are going to be mixed in with a small amount of the recipient's serum, where the antibodies are. By exposing the donor's HLA antibodies to the recipient's serum, scientists can determine if the recipient has antibodies to any of the donor's HLA. This will help us understand if you as the recipient, have antibodies that will attack the donor organ. A positive cross-match means that the reaction occurred, and that would indicate that the recipient's body will likely reject the implanted kidney and the transplant cannot be confirmed. What we're looking for is instead, kind of like when you hear a negative test for cancer, you're looking for a negative test here. We want no reaction to happen between the donor and the recipient's samples. If that occurs, then that means you don't have any antibodies that will attack this piece of tissue, and therefore you can be mostly assured that the patient will not reject the implanted kidney and that the transplant can be performed. Again, this is complicated genetics, but you can notice that a lot of the same vocabulary, antigens, leukocytes, they all pop up over and over again. So what you're going to do in their next activity is, now that you have the blood typing to narrow down who could be a potential donor, you're going to be looking at the tissue typing to see which of those potential donors would be even more optimal for the transplantation of kidney tissue in our example. Good luck.